In this class, we discussed about the next unit, immunotechnology. In this unit, we discussed hybridoma and monoclonal antibodies, hybridoma technology and production of monoclonal antibodies, antibody engineering using genetic manipulation, alternative to hybridoma technology, production of human and humanized antibodies, and uses of monoclonal antibodies. Antibodies are immunoglobulin or protein molecules produced by a specialized group of cells called B lymphocytes, and antibodies are a part of the defense system to protect the body against the invading foreign substances, namely antigens. Each antigen has specific antigen determinant located on it. It is called as epitopes. The antibodies have complementary determining regions which are mainly responsible for the antibody specificity. In response to an antigen, B lymphocytes gear up and produce many different antibodies. These type of antibodies which can react with the same antigen are designated as polyclonal antibodies. The polyclonal antibody production is variable and is dependent on factors such as epitopes, response to immunity. Due to lack of specificity and heterogenic nature, there are several limitations on the utility of polyclonal antibodies for therapeutic and diagnostic purpose. And monoclonal antibody is a single type of antibody that is directed against a specific antigen determinant, that is epitope. It was a dream of scientists to produce monoclonal antibodies for different antigens. In the early years, animals were immunized against a specific antigen. B lymphocytes were isolated and cultured in vitro for producing monoclonal antibodies. This approach was not successful since culturing normal B lymphocytes is difficult and the synthesis of monoclonal antibodies was short lived and very limited. And it is interesting that immortal monoclonal antibody producing cells do exist in nature. They are found in the patient suffering from a disease called multiple myeloma, a cancer of B lymphocytes. It was in 1975. George Kohler and Caesar Milstein got Nobel Prize in 1984, achieved large scale production of monoclonal antibodies. They could successfully hybridize antibody producing B lymphocytes with myeloma cells in vitro and create a hybridoma. The result is that the artificially immortalized B lymphocytes can multiply indefinitely in vitro and produced monoclonal antibodies. The hybridoma cells possesses the growth and multiplying properties of myeloma cells but secrete antibody of beta lymphocytes The production of monoclonal antibodies by the hybrid cells is referred to as hybridoma technology. Next one is hybridoma technology and production of monoclonal antibodies. The myeloma cells used in hybridoma technology must not be capable of synthesizing their own antibodies. The selection of hybridoma cell is based on inhibiting the nucleotides synthesizing machinery. The mammalian cells can synthesize nucleotides by two pathways, that is de novo synthesis and salvage pathway. The de novo synthesizing of nucleotides requires tetrahydrofolate, which is formed from dihydrofolate. The de novo synthesize the nucleotides requires tetrahydrofolate, which is formed from dihydrofolate. The formation of tetrahydrofolate can be blocked by the inhibitor aminopterin. The salvage pathway involves the direct conversion of purine and pyrimidine into the corresponding nucleotides. Hypoxanthine guanine phosphoribosyl transferase, short form is HGPRT, is a key enzyme in the salvage pathway of purine. It converts hypoxanthine and guanine respectively to inosine monophosphate and guanosine monophosphate. Thymidine kinase, short form is TK, involved in the salvage pathway of pyrimidines, converts thymidine to thymidine monophosphate, short form is TMP. Any mutation in either one of the enzymes, that is HGPRT or TK, blocks the salvage pathway. When cells deficient in HGPRT are grown in a medium containing hypoxanthine aminopterin and thymidine, that is HAT medium, hypoxanthine aminopterin and thymidine, that is HAT medium. They cannot survive due to inhibition of de novo synthesis of purine nucleotides. Thus, cells lacking HGPRT and grown in HAT medium is die. And the hybridoma cells possesses the ability of myeloma cells to grow in vitro with a functional HGPRT gene obtained from the lymphocytes. Thus, only the hybridoma cells can proliferate in HAT medium, and this procedure is successfully used for their selection. And the next topic is production of monoclonal antibodies. The basic protocol for derivation of monoclonal antibodies is the production of monoclonal antibodies involves the 6 different steps. That is, first one, immunization, cell fusion, third one selection of hybridomas, and fourth one screening the products, and fifth one cloning and propagation, and sixth one characterization and storage. And first one is immunization. The very first step in hybridoma technology is to immunize an animal with appropriate antigen. The antigen, along with an adjuvant like fluent's complete or incomplete adjuvant, is injected subcutaneously. Adjuvants are non-specific potentiator of specific immune responses. The injections at multiple sites are repeated several times, and this enables increased stimulation of B lymphocytes, which are responding to the antigen 3 days prior to killing of the animal. A final dose of antigen is intravenously administered. The immune stimulated cells for synthesizing of antibodies have grown maximally. By this approach, the concentration of the desired antibodies is assayed in the serum of the animal at frequently intervals during the course of immunization. When the serum concentration of the antibodies is optimal, the animal is sacrificed. The spleen is aseptically removed and disrupted by mechanical or enzymatic methods to release the cell. The lymphocytes of the spleen are separated from the rest of the cells by density gradient centrifugation. And the next step is cell fusion. Thoroughly washed the lymphocytes are mixed with HGPRT defective myeloma cells. The mixture of cells is exposed to polyethylene glycol for a short period. Since it is toxic, polyethylene glycol is removed by washing and the cells are kept in fresh medium. These cells are composed of a mixture of hybridomas, free myeloma cells, and free lymphocytes. And third step is selection of hybridomas. When the cells are cultured in hat medium, only the hybridoma cells grow while the rest will slowly disappear. This happens in 7 to 10 days of culture. Selection of a single antibody producing hybrid cells is very important. This is possible if the hybridomas are isolated and grown individually. The suspension of hybridoma cells is so diluted that the individual aliquots contain on an average one cell each. These cells, when grown in a regular culture medium, and fourth step is screening the products. The hybridomas must be screened for the secretion of the antibody of desired specificity. The two techniques, namely ELISA and RIA, are commonly used for this purpose. In both the assay, the antibody binds to the specific antigen, and the unbound antibody and other component of the medium can be washed off. Thus, the hybridoma cells producing the Desired antibody can be identified by screening. The antibody secreted by the hybrid cell is referred to as monoclonal antibody. And fifth step is cloning and propagation. The single hybrid cell producing the desired antibody are isolated and cloned. Two techniques are commonly employed for cloning hybrid cell: limiting dilution method and soft agar method. First one, limiting dilution method. In this procedure, the suspension of hybridoma cells is serially diluted and the aliquots of each dilution are put into microculture wells. The dilutions are so made that each aliquot in a well contains only a single hybrid cell. This ensures that the antibody produced is monoclonal. And second one is soft agar method. In this technique, the hybridoma cells are cultured in soft agar. It is possible to simultaneously grow many cells in semi-solid medium to form colonies. These colonies will be monoclonal in nature. In actual practice, both the above techniques are combined and used for maximal production of monoclonal antibodies. And sixth one is characterization and storage. The monoclonal antibody has to be subjected to biochemical and biophysical characterization for the desired specificity. It is also important to elucidate the monoclonal antibody for immunoglobulin class or subclass, the epitope for which it is specific and the number of binding site is possesses. The stability of the cell lines and the monoclonal antibodies are important. The cells must be characterized for their ability to withstand freezing and thawing. The desired cell lines are frozen in liquid nitrogen at several stages of cloning and culture. And next topic is large scale production of monoclonal antibodies. The production of monoclonal antibodies in the culture bottles in rather low, the yield can be increased by growing the hybrid cell as assays in the peritoneal cavity of mice. The ascitic fluid contains about 5 to 20 milligram of monoclonal antibodies per ml. This is far superior than the in vitro cultivation techniques, but collection of monoclonal antibodies from ascitic fluid is associated with the heavy risk of contamination by pathogenic organisms of the animals. In addition, several animals have to be sacrificed to produce monoclonal antibodies. Hence, many workers prefer in vitro techniques rather than the use of animals. And encapsulated hybridoma cells for commercial production of monoclonal antibodies. The yield of monoclonal antibodies production can be substantially increased by increasing the hybridoma cell density in suspension culture. This can be done by encapsulating the hybridomas in alginate gels and using a coating solution containing polylysine. These gels allow the nutrients to enter in the antibodies to come out. By this approach, a much higher concentration of monoclonal antibodies production can be achieved. That's about the production of monoclonal antibodies. Thank you.